Hello there, welcome once again to our science lesson. Remember, we have been through this journey of sense organs, and their role is to respond to changes in the environment. It is important that the body responds to change in the environment so that it escapes maybe dangerous environments, such as that if, for example, we have maybe very high temperature that could denature the enzymes, is supposed to escape that. So we have mentioned several parts of the body that are concerned with responses. So we are still moving on. The other structure is the tank. Now, before we look at the tank as a response organ, we can also try to review some other functions of the tank. Remember, we came across the tank in the digestive system. The tank forms a very integral part of the digestive system. It is found in the mouth. But in the mouth, for digestive purposes, the tank performs some other functions. One of the functions of the tank is to turn the food in the mouth during chewing. Turns the food in the mouth. When chewing. That one we know. Suppose you have a problem with your tongue, it becomes very difficult for you to eat because the process of turning food becomes a problem. Then as it turns the food in the mouth, it also mixes the food with saliva, which is again just the same process of turning. But very important, after turning the food during the chewing process, it rolls food substance into boluses for easier swallowing. Into boluses for swallowing. Now, as food is swallowed, it's not just swallowed in an amorphous shape. There are small balls of food. We call them boluses. So those boluses are usually results from the activity of the tongue rolling the food into the boluses. Now, apart from that, again, the other role of the tongue. Right now, I'm communicating to you. I'm producing different kinds of sound because of the tongue. We have the larynx or the voice box, which produces sound, but that sound could be uniform. It's the role of the tongue now to produce different words. So the tongue, apart from other functions that we shall mention in relation to response, it is involved in communication. Involved in communication. So the speech that we make is due to activity of the tongue in the mouth, pushing up, down the sounds, sideways, back and forth, and in the process, words come out. And then the fourth function that is in relationship to our topic, response, is detecting different types of tastes. So before you swallow that food, you already know if it is sweet, if it is sour, if it is too much, if there's too much salt, if there is less salt because of the tongue. So in this lesson, we are going to find out how is it possible that the tongue is able to perform this activity of tasting food. So in other words, the tongue is a sense organ. A sense organ for taste. We know that there are different types of tastes. So the food you eat could be either sweet. You can't tell that the food is sweet just by looking at it. You can tell that by dropping small particles of the food on your tongue. Like, for example, if it is tea or coffee, you'll add in sugar and it becomes sweet. Your food may be salty, because again the tongue is able to detect that. It may be sour, like milk. Fermented milk is sour. Or it could be bitter. Bitter, for instance, pepper. We know that pepper is bitter, or acetic acid. So I'm saying the tongue is able to taste all these: sweet, salty, sour, bitter taste. But we have different types of tastes that are intermediate, either in between sweet, salt, sweet, sour, sweet, bitter. Thousands of them. We have different types. Thousands of tastes exist. So we cannot just narrow ourselves that we are going to have sweet, salt, sour, bitter taste. We have some tastes that will overlap with each other. So the question is, how is it possible that the tongue is able to taste all this? Then it must be having some specialized structures for that purpose. So the tongue is able to detect different tastes, as we have already mentioned. But the structures that perform that role are the taste buds. So it means the tongue has taste buds that are able to detect different types of tastes. So if this is the tongue, a very simple structure of the tongue, this part here the tip part of the tongue usually is able to taste salts. But now we have another part there just close to the tip, is the one that is able to taste for sweet taste, and we have another part, that point, that tastes for sour taste, and just at the back of the tongue it has the ability to taste bitter, or a bitter taste. But it usually overlaps. It's not a must that point it stops there. There is an overlap in between there. So you realize that sometime, even if you drop there some sugary substance, it is going to taste sweet. Or if you drop some salt substance at that point, it's going to taste salty. So these different parts of the tongue are able to do that because they have taste buds. But these taste buds are usually found in a groove. So if we pick this part of the tank, there'll be existence of a groove. We do cross section of the tank. So in this groove, we have cells. We have cells. So those cells that are able to detect different tastes are the ones that we call taste buds. And if you look at all these, salt, sweet is sugar, sour is maybe lactic acid, bitterness could be acetic acid. They are able to taste chemicals. So chemicals are the one that gives rise to different types of tastes. And because taste buds are able to taste chemicals, they are said to be chemoreceptors. So taste buds are chemoreceptors. Why? Because they are able to detect different types of chemicals. But I'm saying these taste buds are found in a groove. So this is a groove. There's a groove here. And in this groove, that's where we shall get the taste buds. But if we pick one taste bud and maybe look at its structure, this is how it will appear. So I pick one there. This is how it will appear. So the taste buds are made up of sensory cells. These sensory cells usually are joined to sensory neuron. Sensory neuron. And then in this part we have hairs. And then there's a pore here called taste pore. Taste pore. And then we have supporting epithelial cells. Supporting epithelial cells. So what will happen is this. When food gets into the mouth, we have saliva. Remember, the role of saliva is to dissolve. The food will be dissolved. When it is dissolved, it will get into the groove. So we have food here. But it will get in touch with the taste buds. Now, when it gets in touch with the taste buds, it's going to stimulate the hairs in the taste pore. And those hairs in the taste pores will again lead or make the sensory cells to be stimulated, and in the process, there will be generation of a nerve impulse. That nerve impulse will be transmitted to the part of the brain that is involved for testing. That part of the brain, we were able to identify it as cerebrum. So in a nutshell, we are saying taste buds are found in grooves. Those grooves are found on the surface of the upper part of the tongue. In those grooves, we have these cells, those chemoreceptors. This is the structure of the chemoreceptors, which are the taste buds. It's made up of sensory cells. But those sensory cells have hairs. The hairs are just meant to increase surface area so that the sensory cells can be able to detect the taste of the food very fast. And then we have supporting epithelial cells. So in words, we can say taste buds are found in grooves on the upper part of the tongue. On the upper part of the tongue. The buds are made of sensory cells that have hairs in the taste pore. So, for sensation to be arrived at, we have said food will dissolve in saliva. Food dissolves in saliva, and then it will diffuse and diffuses into the grooves. Diffuses into the grooves. So while in the grooves, what will happen? It will further diffuse diffuses to taste pores. Hairs are stimulated. Remember, hairs of the sensory cells. Hairs are stimulated to generate a nerve impulse. Generate nerve impulse, which will now be transmitted, that is transmitted to cerebrum. Cerebrum, part of the brain, for interpretation. For interpretation. So a simple test just to find out an experiment. You can also always do these experiments by yourself. Just say this is experiment 2. The aim of the experiment to determine the parts of the tongue. To determine the parts of the tongue that are responsive to different tests. So this is what you will require. Requirements. You need this requirement to work in pairs. To work in pairs, which means you can't do the experiment without a friend. You need to work with a friend. Or some second party. Then you'll require 4 solutions. For solutions in a beaker. So each solution in a beaker or in beakers. 4 solutions in beakers, and they'll include the following: salt solution to give us salt test, sugar which will give sweet test, acetic acid, to give sour taste, or you can use fermented milk, and quinine. You can take quinine tablets to represent bitter tastes. Then requirement number 3, you'll need dropper. So the procedure. So you're going to have one person being the experimenter, and then you yourself, you can be the subject. Subject means the 3 solutions or the 4 solutions will be administered on your tongue. Then we shall have the experimenter who will be dropping the solutions on your tongue. So the first one, experimenter to draw 4, experimenter to draw 4 outlines, of the tongue. outline of the tank on a piece of paper. So we can do this with you for outlines. So the first part, you can have that part. So mention this one. This is the same as that. Number 2, this point is the same as this. Number 3, and then 4 on a piece of paper. Number 2, then let the subject, procedure number 2. I hope this is clear. So you go to the next procedure. If you are making a note, it's very easy. You can always rewind and get it. Number 2 procedure. Let the subject stick out his tongue. Stick out his or her tongue. As far as possible. As far as possible. So you let the subject to stick out the tongue so that nearly all the regions that are meant for testing can be accessed. Then 3, draw a small amount of one of the solution, of one of the solution, amount of one of the solution. And place a drop of it, and place a drop of it on the subject's tongue. On the subject's tongue. But you should also ensure that the subject is not able to know the solution that has been drawn. Let the subject not know the solution. So for this case, you can ask the subject to close the eyes during the experiment. So could be closing with closed eyes. Now, we are going to have 2 responses here. If the subject is able to test the solution, let him or her nod the head in an agreement that he has tested or she has tested. If she does not test anything, let her remain without or he should not nod his head. So if the subject can test the solution, the solution, he should nod. He should nod. He should not nod his head. This is an agreement that indeed I've tested what you have put on the tank. Then 5, for that is procedure 4. Procedure 4, repeat the same with other parts of the tank. Repeat the same with other parts of the tank. Now you'll pick a different solution. You picked maybe a sweet test, sugar test, or sugar solution. You go to salt, you go to quinine, you go to acetic acid. Then 5, every time a drop is placed on the tank, is placed on the tank. Every time a drop is placed on the tank and the subject nods his head or her head, put a tick into the region, a region where the drop was placed, or a solution. So if that's part of the, and then you dropped here a solution. You dropped at that point a solution of salt, and he nods. He does not know that he's salt, and he notes that he has tasted something. There's something. He does not say anything. He has tasted. So you can put a tick, or you can write on your paper salt. Then you'll move to another area. You'll move to another area. Maybe that point. Then you say, you drop again. You have dropped, he has noted his head. But to you, you know, on a paper somewhere else, you'll say sweet. And then you move on to other parts of the tank. You drop here again. He nods the head. It is you who knows why he's nodding, because he has tasted something sour. So you can put there the word sour. Then you move behind the tank. You drop their solution, and the subject again nods. He does not know, but he has tasted something. He has not told you, but it is you who knows that that is bitter. So in this way, you'll have developed a map of the tank showing different parts of the tank, testing different kind of flavors or different kind of tests. So this is a simple experiment. You can always do it even at home with somebody else. Now, why is it that testing is very important? Importance of test pads or testing. Now, if for example you are eating, you have gone to a restaurant, you have bought some food, and then you know the kind of taste that you expect from certain kind of foods, but it comes out very different. You can be able to tell that that food is spoiled. If the food is spoiled, it could be having bacteria, which can easily affect your body system. It can allow microorganisms get into your body system. So for that purpose, testing is important, because you are able to tell the kind of food you are eating, you are able to tell the kind of test that you wanted, and then at the same time, you are trying to prevent the body from maybe getting injuries. Because like a test, for instance, a bitter test, or strongly sour test, or strongly scorching test, that shows that the food that you want to eat can, even if it goes into the digestive system, it is going to damage some organs in your body. So one major function or role or significance of testing is to protect the body from otherwise harmful substances. If, for example, you are taking food and it has a lot of salt, it's not good food for you, because if that food lands into your stomach, it's going to affect the ionic balance. You will require to take in a lot of water. Sometimes a lot of water will be drawn from the stomach cells or the digestive tract. In the process, it is going to offset what you call osmotic balance. So it will be protecting the body against those harmful substances. Acetic acid, for instance, if it is taken in huge quantities, again, it is a problem. Even the sugar that is sweet, too much of it, again, it is not good for our body. So the body will be being protected from those kind of harmful substances. So to recap on what we have gone through, let us look at the following questions. So one of the questions is with study the diagram below and label the parts. So this diagram is showing part of the tank. So we have the parts that is labeled: part A, part B, C, and then D. So when you are labeling, label the parts to show different. I didn't finish. To show different test regions of the tank. So the part that is in front, the posterior part of the tank, it has which test? Salt. It will test salt. Then B, sweet test. C, sour test. And D, bitter test. The other question, question 2, Complete the sentences below. Part A of the sentence, test pads are and detect chemicals. Test pads are dash and detects chemicals. So we have said test pads, they usually test chemicals. So those cells that test chemicals, we have said they are given this name, chemoreceptors. They are chemoreceptors. B, test pads at the back of the tank detect dash tests. Test pads at the back of the tank detects what type of test? We have already given that name, beta tests. C, test pads are found in test pads are found in dash on the upper surface of the tank. We have already said that test pads are cells which are chemoreceptors, but they'll be found in grooves. Found in grooves. In grooves on the upper part of the tank. Nearly all acids give a dash test. In our experiment, we have used acetic acid to show that it can give beta tests. Then E, the flavor of foods is a combination of both. The flavor of food is a combination of both test and dash. Now for you to get how sweet, how bitter, how good the food is, if it is very nice, you shall have to test it first, and also the nose will come in. So smell. The flavor of food is a combination of both the taste and its smell. The olfactory sense comes in to help. Then the last question, the tongue also has receptors that detect, and that is part F. The tongue has receptors that has also, the word also is important. We have mentioned some tests above. Also receptors that detect dash and dash. We have mentioned beta, so the remaining is going to be salt, sweet. So we have bitter, salt, sweet, and even sour. Then an assignment that you'll do on your own, draw a diagram to show the structure of test pads and label it clearly. Mebaki ngapi ba? Nianzishe kitu ingine. Ngapi? 14. Naanzisha something nimalize. The nose. Now, we have gone through the tongue, a very important responsive organ. The other one is the nose. In the nose, we have chemoreceptors that detect smell. We call those chemoreceptor cells as olfactory cells. So has olfactory cells. Olfactory cells. Nose has olfactory cells. These olfactory cells, just like the taste buds, are chemoreceptors. And we have said chemoreceptors are those cells that are able to detect chemicals. Therefore, detect chemicals. So in essence, the nose and the taste buds or the tongue, they work hand in hand because both of them are able to detect presence of chemicals in the system. Chemoreceptors are present in the nasal cavity, and also chemoreceptors are present on the tongue. But we have to look at the structure of the olfactory cells. Structure of olfactory cells. So this structure involves sensory cells, or consist of sensory cells, these sensory cells, they stick out of the epithelial layer of the nasal cavity, but it has some hairs. But now they are supported, those cells, this is the cell we are talking about, it is supported by mucus secreting cells. So we shall have mucus secreting cells at that point, which will now secrete mucus. So this is a mucus layer. This is the real cell, sensory cell in the olfactory system. Sensory cell, that is sensory neuron. Sensory neuron, but it has supporting supporting mucus cells. Supporting mucus cells. So they secrete this layer of mucus, but it has hairs. Hairs. Those are the hairs. And then above this point here is the cavity. Nasal cavity. So what will happen for detection to happen? Air is going to carry chemicals, maybe in form of air particles, molecules. They'll be in form of air particles. They'll get into the nasal cavity during inhaling. So there are two ways, either through inhaling or sometimes through the mouth. When the tongue is swallowing, it pushes up the upper roof of the pharynx, and some of those particles get into the nasal cavity. So there are two entries to the nasal cavity, through the nose or through the mouth. During swallowing, the tongue will push up, and some of the particles in food will reach the nasal cavities or will reach these olfactory cells. So those air particles containing chemicals will dissolve in the mucus layer. It will dissolve in that mucus layer. So it will be converted from air phase or gas phase into liquid phase. When that one happens, because mucus is in contact with the hairs of the sensory cells for olfactory cells, is going to be detected by these hairs, and then it will be stimulated. Sensory cells will be stimulated in the olfactory system. A nerve impulse will be generated. It will be transmitted to the brain. But specific part of the brain where this happens is olfactory bulb. Where is olfactory bulb? Olfactory bulb is in the cerebrum. So remember, all tasting, all smelling is found in this, all vision, hearing, touch. Those five senses are usually found or detected by the part of the brain called cerebrum. So once that message, sensory message, goes to the olfactory bulb in the cerebrum, then one can tell the type of smell that one is exposed to. And it's very important that smell happens. We want to find out the role of smelling. But you realize that there is this olfactory system is very adaptive. You can get into a smoky room, not too much, but let us say moderate. The first time you get into the room, you realize that it is too strong. But as you continue sitting in the room, the olfactory cells become adapted to that condition. And the strong smell that was there initially usually fades with time. That's why some smells or some different smells can only happen, or you only detect them when they are new to you. But once you get into that environment, it becomes like there was nothing, or there was no smell at all, unless it is a very strong smell of a given substance. So what is the importance of animals detecting smell? Significance. Significance of detecting smell. In animals. Now, if you get into a room and you realize that there is a leak of methane gas. Methane gas is not it's not poisonous, yes, But suppose there is an ignition somewhere, a matchbox or something, it can easily cause fire. That's why the methane gas, the cooking gas, has that smell. It is then the manufacturer's responsibility who has put there the smell, so that if there is a leakage of the gas, your olfactory system is able to detect it, so that you can go to the point of leakage and close it. So it is important because it's going to protect you from harmful substances. So one of them is protection. For protection from harmful substances. We also have poisonous gases. Poisonous gases like hydrogen sulfide, sulfur 4 oxide, chlorine gas. The olfactory cells are the one that will detect that actually there's a problem, and then you'll be able to find the best way of saving the situation. So another role is finding suitable food. It helps animals to find suitable food. For human beings, you'll always want food that has good aroma, some good smell. And for wild animals, like a lion, to detect where the antelope is going to use the olfactory system or olfactory sense, so that it can detect that this direction or in this direction we have an antelope, and not going the direction where we have an elephant, which is very difficult to kill. So it will rather go to the suitable food, the antelope, finding suitable food. And then another one, detecting predators. For detecting predators. This is very useful again in the jungle or in the wild. If we have an antelope at that point, and a lion in this direction, and the wind is coming in this direction, the antelope can detect that there is a predator there. So in the process, it can find a way of moving away so that the lion cannot get it. Otherwise, it will be preyed on. So detecting predators. But at the same time, if it was the wind is going this direction, the lion will be able to detect the antelope, which is suitable food material for the lion. So it is important for survival in the jungle. Even finding mates, important in finding mate. Sexual partner, finding mate through the smell. The pheromones are released, but they are picked through the nose. We have those chemicals, pheromones, are released, especially, for instance, the dogs will release this. Female dog is going to release these pheromones into the air, and then they'll diffuse through air to male dog or dogs, and then they'll be attracted to the female dog, especially when it is ready for mating. So finding the mate. I think those are the major importance. At your own time, you can also, through the internet, find several other significance. Then just the very last question, a question as we wind up. Question. Explain why when one has flu he is not able to detect the flavor of food readily. So when you have flu, very common symptom of flu, it comes with running nose. Running nose. In other words, there'll be high rate of secretion of mucus. There's buildup of mucus in the nasal cavity. When there's buildup of mucus in the nasal cavity, remember, as we have been able to describe the structure of the olfactory system, what will happen? There will be a thick layer of mucus covering these hairs. So once we have a thick layer of mucus covering these cells, then it will be very difficult for those molecules or chemicals to dissolve through this mucus lining to reach the hairs for detection and stimulation, so that a nerve impulse is sent to the brain to show that actually we have this kind of flavor or taste. So that's why when you have flu, because of the running nose or high concentration of mucus in the nasal cavity, it blocks the hairs. And when the hairs are blocked, there will be no stimulation to generate an impulse for detection of different kind of smell. So we can stop there. In our next lesson, I think we shall be comparing the different kind of responsive organs. Welcome.